Hello everyone, welcome to my Medication Academy channel. In May 2025, the US FDA approved 3 novel chemical entity drugs. One is a combination of small molecule oral targeted therapy for treating low-grade serous ovarian cancer. One is a RV injectable antibody drug conjugate for the treatment of non-small cell lung cancer. The third one is a topical eye drop therapy for treating dry eye disease. Now, let's dive into each medication in more detail. Avutometinib, defactinib, brand name AVMAPKI FAKZYNJA Co-Pack. This combination medication products was approved by the US FDA on May 8, 2025, and indicated to treat KRAS-mutated recurrent low-grade serous ovarian cancer, LGSOC, in adult patients who have received prior systemic therapy. This combination therapy product contains 2 medication products. One, AVMAPKI, is available as a 0.8 mg capsule. FAKZYNJA is available as 200 mg tablets. For recommended dosage, avutometinib 3.2 mg, which is 4 capsules, taken orally twice weekly on day 1 and day 4. For defactinib, 200 mg, which is 1 tablet, taken orally twice daily for the first 3 weeks of each 4-week cycle, which means for this recommended dosage therapy regimen, so you can see patients are on this combination therapy for 3 weeks and then off for 1 week. This dosing regimen would help mitigate the adverse effects for this combination products, in particular related to MEK inhibitor. This combination therapy product contains 2 drug molecules. For avutometinib, it's a small molecule, molecular weight 471.46 gram per mole. Hydrogen bond acceptor 10. Hydrogen bond acceptor refers to the number of oxygen and nitrogens within the drug molecule. Hydrogen bond donor 2. Hydrogen bond donor refers to the number of hydroxy and NH groups within the drug molecule. Log P 1.34. Polar surface area 141 square angstrom. This drug molecule is an acidic drug molecule for this NH group because of the electron withdrawing effect from sulfur group as well as from the fluorine. So therefore this drug molecule is formulated as a potassium salt. So this would be deprotonated, negatively charged. So it's available as a potassium salt. For defactinib, molecular weight is 510.50 gram per mole. Hydrogen bond acceptor 11. Hydrogen bond donor 3. Log P negative 0.74. Polar surface area is 150 square angstrom. This drug molecule is available as hydrochloride salt form. Mechanistically, avutometinib ending with metinib, so this drug molecule indicating this is a member of MEK1 inhibitor class, works as a mitogen activated protein kinase 1 MEK1 inhibitor. For defactinib, it's an inhibitor of focal adhesion kinase FAK and protein-rich tyrosine kinase 2 (PIK2). Pharmacokinetically, for avutometinib, Tmax about 2 hours under fasted conditions. For defactinib, it's about 4 hours under fed conditions. Half-life for avutometinib, 51 hours. So you can see very long half-life. For defactinib, half-life is about 9 hours. So therefore, these two medications, they have a very different half-life. So therefore, this is part of the reason. So you can see for avutometinib, it's taken twice weekly, but for defactinib, it's taken twice daily dosing because they have very different half-life profiles. So therefore, they need to take this combination products separately. So therefore, they cannot co-formulate this one as one combination product because of the very different pharmacokinetic profiles. Protein binding for avutometinib, 99%. For defactinib, about 90%. Volume of distribution at the steady state for avutometinib, 25 liter. For defactinib, 1,560 liter. Metabolism, so you can see both medication, they are the subject for CYP3A4. In addition, for defactinib is also subject for CYP2C9. Excretion profiles in the urine for avutometinib, 52%, but 3.2% as the parent drug molecule. In the feces, 39%, but 9% as the parent drug molecule. For defactinib, in the urine, 7.6%, but 0.8% as a parent drug molecule. In the feces, 87% with 52% as a parent drug molecule. As mentioned, given both medication, they are subject for CYP3A4. So therefore, with regard to drug-drug interactions, for example, strong and moderate CYP3A inhibitors, for example, the azole and fungal agents like itraconazole. So this would increase the drug exposure for both medication products. So this would increase the risk of the adverse effects. So therefore, in this case, would avoid concomitant use. Similarly, for patients, if they take strong and moderate CYP3A inducers, for example, phenytoin, in this case, the inducers would make the medication metabolize much faster. So this would lower the drug exposure for this combination product. So therefore, this can lead to decreased efficacy. Therefore, in both cases, strong and moderate CYP3A inhibitors or inducers need to avoid concomitant use. For patients, if they take some other, for example, anticoagulants, warfarin, this would increase the drug exposure for warfarin. So therefore, this can lead to increased risk of bleeding. Avoid concomitant use. If unavoidable, so make sure monitor RNR for warfarin closely. For patients, if they also take gastric acid reducing agents, for example, PPI, proton pump inhibitor, or H2 blockers, because the gastric acid reducing agent, this would increase the pH value, and this would decrease the absorption or solubility for defactinib. so therefore require avoid concomitant use. If unavoidable, so make sure take this medication products FAKZYNJA 2 hours before or 2 hours after the gastric acid reducing agent. For adverse effects, at least 25%, nausea, fatigue, rash, diarrhea, musculoskeletal pain, edema, vomiting, abdominal pain, dyspepsia, dermatitis, acneiform, vitreoretinal disorders, stomatitis, that's the inflammation of the mouth, pruritus, visual impairments, constipation, dry skin, dyspnea, cough, urinary tract infections. Telisotuzumab, vedotin, brand name EMRELIS. This medication was approved by the US FDA on May 14, 2025, and indicated to treat adult patients with locally advanced or metastatic non-squamous non-small cell lung cancer with high c-MET protein overexpression for patients who had prior systemic therapy. This medication is available as a 20 mg or 100 mg as a lyophilized powder for RV intravenous use. Recommended dosage, so you can see in this case, it's based on the patient's body weight based dosing, 1.9 mg per kilogram, RV infusion, over 30 minutes every 2 weeks. As you can see, based on this drug molecule, this is a member of ADC drug class, antibody drug conjugate. You can see this medication contains this mAb along with the drug payload MMAE. So that's the small molecule payload MMAE. So that's the microtubule inhibitor, along with the linker and the spacer in between. Given this medication product contains monoclonal antibody, so therefore the drug product need to be stored refrigerated 2 to 8 Celsius. Mechanistically, this c-MET directed antibody drug conjugate binds to the c-MET cell surface receptor for hepatocytes growth factor. But in general, for this class, ADC drug class, antibody drug conjugates, they bind with the receptor, kind of overexpress the receptor on the cancer cells. Once bound with the receptor, in this case the c-MET, and then the drug complex will be internalized within the cancer cells, and later on the small molecule will be released, microtubule inhibitor MMAE. So that's the monomethyl aristatin E drug molecule. So that's a microtubule inhibitor. By this protein-lytic cleavage. So therefore, releasing this small cytotoxic MMAE payload small molecule, this would lead to the cell cycle arrest and apoptosis, programmed cell death. Pharmacokinetically, volume of distribution 3.4 liter, half-life 3 days for this ADC compound complex, like antibody drug conjugate, but 4 days for this unconjugated small molecule payload MMAE. Protein binding based on in vitro studies ranging from 68 to 82%. Metabolism. This medication product is metabolized, expected into small peptides and amino acid and unconjugated MMAE small molecule, so once cleaved from the antibody complex. In addition, this small molecule MMAE is a subject for CYP3A4 enzyme. Another thing I want to mention, this ADC drug conjugates, so you can see there's a 3, so that's referred to drug antibody ratio, which means one antibody is attached to approximately 3 small molecule like MMAE payload. As mentioned, this MMAE is a subject for CYP3A4, so therefore with regard to drug-drug interactions, strong CYP3A inhibitors, this would lead to increased drug exposure for MMAE payload because the drug molecule will be metabolized slowly. So therefore, concomitant use may increase the exposure to unconjugated MMAE. So in a similar context for CYP3A inducers, so this can lead to decreased exposure to unconjugated MMAE. For adverse effects, at least 20% peripheral neuropathy, fatigue, decreased appetite, peripheral edema. Acoltremon, the brand name TRYPTYR. This medication was approved by the US FDA on May 28, 2025, and indicated for the treatment of the signs and symptoms of dry eye disease. This is a small molecule with lipophilic 2-isopropyl 5-methyl cyclohexane. So basically this is a lipophilic motif with the carboxamide linker spacer in the middle and 4-methoxyphenol motifs. Molecular weight for this medication 289.42 gram per mole, hydrogen bond acceptor 3, hydrogen bond donor 1, log P 5.02. So you can see in this case log P is over 5. So therefore this is a pretty lipophilic drug molecule. Polar surface area 38 square angstrom. Mechanistically, this medication is considered first-in-class neuromodulator for treating the dry eye disease. It's an eye drop. So therefore you can see the recommended dosage: instill 1 drop in each eye twice daily, approximately 12 hours apart. Again, this is a topical eye drop. It's available as 0.003% of formic solution. So mechanistically, this is considered the first neuromodulator eye drop for treating dry eye disease. Works as a transit receptor potential metastatin 8, that's a TRYPM8 thermal receptor agonist. This would lead to trigeminal nerve signaling and increase the basal tear production. Although the exact mechanism of action related to dry eye disease is unclear. With regard to pharmacokinetic profiles, so given this topical ocular dosing, the drug exposure in plasma is minimal. So therefore, for drug-drug interactions, no clinically significant drug interactions are expected. Adverse effects, that's about 50% patient population experience this installation side pain. This is all the key reference which I have used to prepare for today's video. FDA Novel Drug Approval for 2025 is a very valuable resource, and all the other 3 medication products information which are shared today based on their respective prescribing information. And all this information documents, they are either available on the US FDA website or they are available on the manufacturer website as well. The physiochemical properties, they are available from the chemspider.com. Finally, I want to thank you all so much for watching. If you enjoyed the video, please consider subscribing for future medication video updates. As always, keep learning and elevating. Thank you so much.